The amount of force your muscle cells will generate will depend on the length of the sarcomeres. So if your sarcomere is already contracted a little bit, well, you're not going to have that much room to do your sliding filament model. You're not going to generate that much force. But on the other hand, if your sarcomere is stretched out too far, well, it might be too hard for actin and myosin to interact if they can't even reach each other. So you might not even get a contraction if the muscle is stretched too far. So there's this slide in the middle area where you're going to have an optimal contraction. Why? Because things are nice and evenly spaced out, giving them lots of room to do this sliding filament model. No, you do not need to know this for your exam. Like usual, stick to the exam review. We want to focus more, though, on the steps of a muscle contraction. And now we're going to finally start at the beginning. And we're going to bring it down to the level of the muscle cell. How do you stimulate it? Well, we know from our previous chapter that we did, chapter 12, that's going to begin with a neuron. Remember, there are neurons specifically designed to tell muscles what to do. Remember, we call them motor neurons. And remember, neurons typically communicate with other cells using their axon terminal. So when we talk about how a neuron stimulates a muscle cell, we're really talking about how a neuron will use its axon terminal to communicate with a muscle cell. And you know some things already. You know the name of the place where a neuron will communicate with another cell. Remember, we call that a synapse. Remember, a synapse is just a place where a neuron will communicate with another cell. And we're still talking about a synapse. But like usual, this name is going to change. We're talking about a specific synapse. We're talking about the synapse where a neuron will talk specifically to a skeletal muscle cell. We call this a neuromuscular junction. It tells you in the name, neuro for neuron, muscular for muscle. Remember, junctions are cellular junctions. They're between cells. So a neuromuscular junction is just a synapse where a neuron will talk to a muscle cell, where a neuron will stimulate a muscle cell. So like usual, we got to go through some anatomy. And that's what you see on this image here. It's showing some anatomy of a neuromuscular junction. Remember, it's just a synapse. You know the three basic parts of a synapse. Remember, one part is the axon terminal of your neuron sending the signal. That's what you see in the picture off to the right. They've zoomed in, and that yellow structure up top, that's the axon terminal of your neuron. Remember, another part to your synapse was the plasma membrane of the cell receiving the signal. In this case, it's the plasma membrane of the muscle cell, skeletal muscle cell. Remember, that's the sarcolemma. But yet again, the name is going to change. When we talk about the plasma membrane of a skeletal muscle cell, specifically at a neuromuscular junction, we call it a motor end plate. A motor end plate is just the plasma membrane of your skeletal muscle cell, specifically when you're talking about a neuromuscular junction. And remember, when your neurons talk to a cell or communicate with another cell, they don't actually have to touch. Remember, there's a space in between. Remember, we call that space the synaptic cleft or the synaptic space. It's still there because this is still a synapse. So these pictures are just showing you a neuromuscular junction is a synapse just like any other. It'll have those three parts. It will have an axon terminal, where you'll have your chemical messenger inside, your neurotransmitter, and you'll release it to help bind receptors on the muscle cell. And so let's begin charting this journey in a muscle contraction. If you wanted to contract, it will begin with an action potential arriving at the axon terminal of your neuron, of your motor neuron. Why? Why do you need that spark of electricity to arrive? Well, that spark of electricity, that action potential, when it arrives at the axon terminal of your neuron, it's actually going to open some voltage-gated channels. When this spark of electricity, this action potential arrives at the axon terminal, it's going to change the charge across the membrane. And when you do, remember, that will affect some voltage-gated channels. In this case, it's going to cause calcium voltage-gated channels to open. And when these voltage-gated calcium channels open, calcium is going to rush into the cell. And when calcium rushes into the cell, that will trigger the release of acetylcholine, which is our neurotransmitter, which is our chemical messenger. So that's the first step. First step is the action potential arrives at the axon terminal, triggering the opening of voltage-gated calcium channels, which will allow calcium to rush into the axon terminal and trigger the release of acetylcholine. Then once you release the acetylcholine, it has to make that journey across that gap in the middle. It has to make the journey across the synaptic cleft. And it's going to do that using diffusion. It's going to go from where there's a lot of them, which is closer to the axon terminal side, to where there's fewer of them, which is closer to the motor end plate side. So in step 2, we just simply say acetylcholine diffuses across the synaptic cleft. It drifts across the synaptic cleft, and then it will bind to receptors on the motor end plate. That's step 2. Step 2, one more time. Acetylcholine diffuses across the synaptic cleft and binds receptors on the motor end plate. And you're still not moving yet. There's still more steps to go. You already know the last steps. We're not even there yet. As soon as the muscle cell gets its signal, gets the acetylcholine to bind its receptors, remember, this is an excitatory neurotransmitter. We know that already. We're going to generate an excitatory postsynaptic potential in our muscle cell, which is going to cause it to depolarize, aka become slightly positive, until it hits threshold, and it will generate its own action potential. And when it generates its own action potential, that action potential will move as well. It will travel along the entire plasma membrane of the muscle cell. Why is that important? Because you remember, parts of the skeletal muscle's plasma membrane dives deep into the cell. Remember, we call that a T-tubule or transverse tubule. So when the action potential travels along the entire plasma membrane, it will even travel down a transverse tubule. Why? This is finally why that triad comes into play. Remember, a triad has a T-tubule, and on either side of it are sarcoplasmic reticulums. There's a sarcoplasmic reticulum sitting right next to the T-tubule. So when that action potential travels down the T-tubule, it's kind of going to zap the sarcoplasmic reticulum. And the sarcoplasmic reticulum is like this one friend. Everybody has this one friend. Who's this one friend? Everybody has this one friend where whenever you scare your one friend, they fart. Just a little bit, just a little bit. Everyone has that one friend, when they get scared, they fart just a little bit. If you don't, maybe it's you. I don't know. But anyway, the sarcoplasmic reticulum is kind of like that one friend. When the action potential zaps it, it's almost like it farts. But in this case, it's farting out calcium. So the action potential, when it travels down the T-tubule, will trigger the release of calcium from the sarcoplasmic reticulum. And once the calcium gets released from the sarcoplasmic reticulum, it's going to travel throughout the entire cytoplasm, looking for one thing. You know what one thing from our sliding filament model. It's going to look for troponin, and it's going to bind to troponin, causing troponin and tropomyosin to move away from those myosin binding sites on actin. And you know the rest. This one slide shows us all the steps to our skeletal muscle contraction. Let's go through them all one more time, slowly. So the first step is that your action potential will arrive at the axon terminal, which will trigger the opening of voltage-gated calcium channels, and calcium will rush into the axon terminal, triggering the release of acetylcholine. That's step 1. Then step 2 is acetylcholine will then diffuse across the synaptic cleft and bind receptors on the motor end plate. And then that will trigger step 3, which is when your muscle cell will depolarize and generate its own action potential, which will travel along the entire plasma membrane, aka sarcolemma, and down a T-tubule to trigger the release of calcium from your sarcoplasmic reticulum. Then step 4, calcium will bind to troponin, causing troponin and tropomyosin to move away from the myosin binding sites on actin. Then step 5, myosin heads will hydrolyze ATP to form the cocked position. Step 6, myosin heads will bind to actin, forming the cross bridge. Step 7, myosin heads will pull actin towards the M line during the power stroke. And step 8, myosin heads will bind ATP to release actin and cycle again and again and again until you've hit maximum contraction. That's how you move. This one slide does it all. Action potential arises at the axon terminal, triggering the opening of voltage-gated calcium channels where calcium will rush into the axon terminal, triggering the release of acetylcholine. Acetylcholine will drift or diffuse across the synaptic cleft and bind receptors on the motor end plate, which will cause the muscle cell to depolarize and generate its own action potential, which will travel across the entire plasma membrane and down a T tubule, where it will trigger the release of calcium from the sarcoplasmic reticulum. And then that calcium will then bind to troponin, causing troponin and tropomyosin to move away from the myosin binding sites on actin, while myosin heads will hydrolyze ATP to form the cocked position. It will bind to actin, forming the cross bridge. Myosin heads will pull actin towards the M line during the power stroke. Then myosin heads will bind ATP to release actin. And that's how you're going to move. Those are all the steps in the generation of a muscle contraction. Luckily, you don't have to think about any of that besides just wanting to move. But that's how you move. But then you got to consider, how do you relax? Turns out you got to do 2 things to relax. One of them is really attacking the first steps. It's attacking acetylcholine. For you to relax, you got to stop stimulating the muscle cell. And you're going to stop that by getting rid of a neurotransmitter. You even know how to do that. Remember, there are 3 ways to get rid of a neurotransmitter. Which of those ways are you going to use to relax your skeletal muscle? Turns out you're going to use enzymatic degradation. You're going to break it down with an enzyme. And we even know the enzyme that we use to break down acetylcholine. It's in the picture. It's called acetylcholine esterase. Acetylcholine esterase will literally break down and destroy acetylcholine. And so if there's no acetylcholine, your muscle cell will not get stimulated and you'll be able to begin relaxing. But to finish relaxing, you got to do one more thing. You got to get rid of calcium. Remember, calcium is allowing your regulatory proteins to move out the way. You have to put them back. So in order for troponin and tropomyosin to go back to binding to actin, you got to get rid of the calcium. How? You put it back into its sarcoplasmic reticulum. And to pump it back, you're using pumps in the membranes of the sarcoplasmic reticulum that require ATP. So it turns out not only do you need ATP to move, because you need ATP for myosin head to form the cocked position and to release actin, you even need ATP to relax. You need ATP to put the calcium back into the sarcoplasmic reticulum. This is why muscles use so much energy. They not only need energy to move, they even need energy to relax. And because of that, we're going to have to spend some time talking about that. We're going to have to spend some time talking about the sources of energy for your skeletal muscles. Turns out there are 3 major sources of energy for your skeletal muscles and your skeletal muscle cells. What are they? They're listed on the slide. There's something called creatine phosphate, anaerobic glycolysis, and cellular respiration. Those are your 3 major sources of energy for your skeletal muscles. Let's go through them one by one. Starting off with the one that gives you the energy for the least amount of time. Turns out you have 3 sources of energy because they all give you energy for differing amounts of time. And the first one I'm mentioning is only going to give you energy for a couple seconds. It's called creatine phosphate. So how's creatine phosphate giving you energy only for a couple seconds? Well, it has to deal with ATP's structure. We got to dig way back to our structure of ATP, way back to chapter 2. Remember, ATP has 3 phosphate groups in it. Remember, it's called triphosphate because it has 3 phosphate groups. And remember, the energy is in the bonds really between the first and second phosphate group and the second and third phosphate group. So let's say you ate a Whataburger cheeseburger, and odds are, if you're at home right now, you probably ate more energy's worth of food than you're using right now. And so your body's going to try to save some of that energy. What your body's going to do is it's going to take ATP and it's going to take creatine. And it's going to take a phosphate. It's going to pop off that third phosphate group and give it to the creatine to